We are now on to the last step of cellular respiration, which is oxidative phosphorylation. And oxidative phosphorylation includes both the electron transport chain and chemiosmosis. And I think it's important to layer on the information kind of step by step to really conceptualize what's happening during oxidative phosphorylation. This is where we are ultimately going to make the bulk of our ATP. Each glucose molecule can produce about 34 to 38 ATP molecules on average. And we, so far, through glycolysis and oxidative phosphorylation and the citric acid cycle, have produced about 4 ATP. So you can see the bulk of our ATP production is going to come in this phase. And so because it is so important, I think it is necessary to kind of take the time to layer on this information. This process of oxidative phosphorylation is also going to occur in the mitochondria. We saw pyruvate oxidation and the citric acid cycle also happen in the mitochondria. And so I've drawn the mitochondria again with the outer membrane, the inner membrane. We have the matrix here. And in the matrix, we have a bunch of NADH and FADH2, these reduced electron carriers that are bringing these electrons to the electron transport chain. Now remember, these electron carriers were reduced from the oxidation of glucose and its intermediates. And the electron transport chain is a series of protein complexes that are embedded in that inner mitochondrial membrane. And there are many electron transport chains in this inner mitochondrial membrane, and I'm just going to draw a few there. So the role of NADH or FADH2 is to bring those electrons into the electron transport chain. So in our discussion on oxidative phosphorylation, our focus is really going to be here in this section of the membrane to see what happens once NADH drops those electrons off to the first protein complex in the electron transport chain. So what I'd like to do is just take this one portion of the membrane and blow it up, see what we can do. As we blow up that region of the mitochondria, we're really focused on the membranes. And so here you can see that we have an outer membrane at the top. And of course, this outer membrane is made up of a phospholipid bilayer, but I didn't want to write out all the phospholipids, so I'm just representing this phospholipid bilayer as a line, the outer membrane. We have the inner membrane here with a bunch of proteins in it that we'll talk about their role in oxidative phosphorylation. Between the membranes, we have this intermembrane space. And then, of course, within the center of the mitochondria, we have the matrix. So again, we have just blown up this one region of the mitochondria and really zoomed in and focused on the detail here. Now, of course, we have these reduced electron carriers in the matrix, and their job, their role in the cell is to bring electrons that were originally gained from the breakdown of glucose and the glucose intermediates during the first 3 stages of cellular respiration to the electron transport chain. So I have provided a reduced electron carrier here that is present in the matrix. And we can see that we have several protein complexes represented in blue on the screen that are embedded in that inner mitochondrial membrane. And these are just a collection of proteins that play a couple roles in oxidative phosphorylation, including electron carriers. And so NADH is going to drop off its 2 electrons that it's carrying to the first protein complex. By dropping off those 2 electrons, it is that NADH has become oxidized, it has lost its electrons, and goes back to the form of NAD+. Now, of course, it has to also drop off that 1 hydrogen ion that it's carrying, and it's going to drop that off into the matrix, and that's just going to join a pool of hydrogen ions that are in the mitochondrial matrix from each of these electron carriers, dropping off their electrons and releasing those hydrogen ions that they're carrying. Now, this NAD+ can go back into the matrix and again gain electrons from the oxidation of glucose in one of its intermediates. Now, this transfer of these 2 electrons is a redox reaction. In this case, NADH is oxidized. It has lost electrons, and this first protein complex is reduced. And through this redox reaction, we know that some energy is released, and we have to remember that that energy is being released every time these 2 electrons are transferred. Now, some energy is going to be released here, but some energy is still going to be associated with those electrons, and those 2 electrons will continue to be transferred, what we say, down the electron transport chain, each time releasing a little bit of energy through that redox reaction. So again, this time through this transfer of electrons, this first protein complex becomes oxidized, the second protein complex becomes reduced. So when we think about the electron transport chain, we can think about the electron transport chain, and we're just going to abbreviate it ETC, as a series of redox reactions. So it's just one redox reaction after another through transfer of electrons. So we have a series of redox reaction, and then each redox reaction, each time we transfer these electrons, we are going to release energy. So we have this release of energy. And this is the same thing that we saw in when we reviewed energy in our foundational information, but now we're applying it to the process of cellular respiration, specifically in the context of the electron transport chain. Okay. So this is, a pretty simplistic view of the electron transport chain, but it's sufficient for what we need to understand in the context of this particular course. These 2 electrons will continue to be passed down the electron transport chain, each time releasing energy, because we have a redox reaction that has taken place. In this case, this protein complex is oxidized, and this protein complex is reduced. Each time we pass these electrons on from one protein complex to another, we are releasing energy until those 2 electrons are passed on to the final electron acceptor, which is an oxygen atom. In your textbook represents this oxygen atom as a half of an O2. When these 2 electrons are finally passed on to an oxygen atom, that oxygen atom can combine with 2 hydrogen ions that are present in the matrix to produce water, one of the low chemical energy products of cellular respiration. Now, oxygen is very important because oxygen has a high electronegativity associated with it. So when oxygen is present, it is going to pull down the electrons through the electron transport chain, each time releasing energy in a redox reaction, because this oxygen atom has a high electronegativity and is attracted to those electrons. Okay, now let's layer a little bit more information onto the electron transport chain. Now, as I mentioned previously, as all of these reduced electron carriers come into the electron transport chain and drop off electrons, they are also dropping off hydrogen ions that they are carrying. And these hydrogen ions are used to create an electrical and chemical gradient across the inner mitochondrial membrane. And we recognize that there are not only hydrogen ions in the matrix on the inside of the inner membrane, but there is also a high concentration of hydrogen ions in the intermembrane. So again, this is a piece of information that we're layering on to the information that we've already talked about from the electron transport chain. Now, we talked about each time these electrons move through a redox reaction, energy is being released. Well, that energy is going to be used to generate and maintain this hydrogen ion gradient that I have represented here. And the way this is going to happen is that the energy is going to be used to transport these hydrogen ions against their concentration gradient from a low concentration in the matrix to an area of high concentration in the intermembrane space. And this is a form of active transport. We are moving hydrogen ions against their concentration gradient, so we are going to need energy for that, and that energy is going to be provided from the movement of these electrons down the electron transport chain. Now, we know that hydrogen ions are charged, so they can't directly move through the phospholipid bilayer. This is not a possibility because they would get stuck in the hydrophobic center of the membrane. So instead, they need a transport protein to move across that inner mitochondrial membrane. And having that transport protein there is going to allow for that movement. Now, the protein complexes not only act as electron carriers in the redox reactions, those transfer of electrons down the chain, but they also act as transport proteins that are going to allow for those hydrogen ions to move across the membrane. And that's why I have my arrow moving through one of those protein complexes. Now, everything that we've seen here, from NADH coming into the electron transport chain, dropping off those electrons, that series of redox reactions that release energy, and that energy used to generate and maintain a hydrogen ion gradient, is all part of the electron transport chain. So previously we said that oxidative phosphorylation is split between the electron transport chain and chemiosmosis. This is all happening during the electron transport chain. And if we go back to our just kind of bullet points of what's happening during the electron transport chain, we can also add that the energy released from the redox reactions is moving hydrogen ions against their concentration gradient. So if we look at the visual again that we're drawing, kind of generating to look at this process as a whole, you can see that no ATP is actually formed during the electron transport chain. We know the bulk of ATP production is going to happen during this phase, is going to happen during oxidative phosphorylation. But the reality is that no ATP is generated during the electron transport chain, which means that all of the ATP is going to be generated during the second part of oxidative phosphorylation, which is chemiosmosis. So let's see what we have here. Okay? We have this concentration of hydrogen ions. And this represents an electrical gradient. These hydrogen ions are charged. It also represents a chemical gradient. And this gradient carries energy with it. And when we have a high concentration of hydrogen ions in the intermembrane space, we know that these ions are constantly moving around, bumping into one another, and they want to spread out into all available space. But we know that they can't cross directly through the membrane. They need a transport protein. And we do have a transport protein for these hydrogen ions in the membrane that will allow them to move from the intermembrane space back into the matrix, and that is called ATP synthase. And that is the transport protein that is in green over on the right side of your screen. So these hydrogen ions will flow down their concentration gradients from an area of high concentration to an area of low concentration through ATP synthase. And this will activate ATP synthase. When ATP synthase is activated, it will phosphorylate ADP. So a phosphate group gets added to that ADP molecule to generate ATP. Now, of course, the phosphorylation of ADP is an endergonic reaction. It requires energy, and that energy is provided by those hydrogen ions moving down their concentration gradient through ATP synthase. And there's a short video in your online module that shows kind of the detail of how that happens. Okay? So this is what we've been waiting for all along in all of our discussions on cellular respiration, is this production of ATP synthesis. And this is specifically happening during chemiosmosis. Now, as we kind of finish up our discussion, it's really important to synthesize this information and see it happen as a complete and entire process. And to do that, I'm going to work a little bit backwards. Okay? We can't make ATP unless we have a hydrogen ion gradient, this electrical and chemical gradient present via the hydrogen ions. We're not going to be able to generate a hydrogen ion gradient unless we have reduced NADH molecules that allow for this series of redox reactions to happen in the electron transport chain. We can't have reduced electron carriers unless we bring fuel like glucose into our cells that we can break down. So each of the processes that we have been through are really an integral part of the process of cellular respiration and are really all dependent on one another. So we can't forget that big picture, that this NAD is actually reduced by oxidizing the food, like glucose, that we have consumed. So I hope drawing this out really helps you visualize what's happening in oxidative phosphorylation, both in the electron transport chain and chemiosmosis.